you for the introduction. So good afternoon, everyone. I'm Gianluca Lombardi, a PhD student at Sorbonne University, and I'm going to talk about our work on models to predict intrinsic and soft disorder for protein sequences. So starting from the basics, we are all familiar with the structure function paradigm, where given a protein sequence, it falls in a well-defined structure, and then this structure enables it to produce the corresponding function. This paradigm, however, does not always hold, as we know, and in many cases, the sequence does not fall in a well-defined structure. Instead, we have intrinsically disordered regions, and in some cases, we have ensemble of protein conformations where we can find intrinsically disordered proteins. So to extend a bit this definition in order to include more cases, we introduce the concept of soft disorder, and it is defined based on different crystallographic structure from the same sequence. So given a single structure, we will have some residues that are structured, while some of them will be missing. And if we consider an ensemble of structures for the same sequence, then we will see that some of these missing residues will appear in the crystal and will undergo a disorder to order transition. And this is what we normally refer to as intrinsic disorder. Now, among those structural residues, we will have some of them that have a high B factor, which explains the displacement of atoms in the crystallographic structures. And when we, again, compare many structures from the same sequence, we will have a union of these high B factor regions, and also those regions that undergo a disorder to order transition will be present as high B factor regions. And we define this union of high B factor regions as soft disorder. Why this concept is important for okay, sorry. So these analyses were run on almost 200,000 crystallographic structures from the PDB, where we clustered the chains together when their sequence similarity was above 95%. And among those structures, we can then define hard disorder, which correspond to the residues that are always missing in all the structures considered, and soft disorder, which corresponds instead to those regions that present uncertainty in the atom positions and that can distinguish between flexibility and static disorder. So positions where atoms freeze that are different between the crystals. And the importance of soft disorder is highlighted by its topological similarity with interface regions. And this is an example where, for a PDB cluster, where we have different structures, and in these different structures, we have different regions corresponding to interfaces with the specific partners and high B factor regions. But when we consider the union, so the soft disorder, for all these structures, we can see that it kind of overlaps with the union of interfaces, where the union of soft disorder is represented in orange and the union of the interfaces is represented in blue. And we also have some missing residues that are never present in any of these structures. So in a more quantitative way, the soft disorder regions and the union of the interfaces follow these similar patterns in terms of the relative size of the two regions, and also in terms of the number of regions with respect to the cluster size, so the number of chains considered. Also, soft disorder is important as it modulates the assembly paths. This is an example for the P53 complex. So when we consider a single unbound structure for the P53, we can see that some regions in this structure present a high B factor, and those regions correspond to the same regions that bind to partners that include both another protein and DNA. When this partner is recruited, then the soft disorder regions change, and we can now find that these new regions with high B factor correspond to new binding regions for other partners. And this can be seen also in other protein complexes. So our question is now, can we predict both intrinsic and soft disorder from the protein sequences? And there have been many methods that have been developed in the recent years for the prediction of intrinsic disorder. And the most recent one, and also most successful, use protein language models as representation for these proteins. So very briefly, protein language models are large transformer architectures that are trained on protein sequences, and usually the learning task is masked language model objective, where some of the residues are masked and they are predicted based on the context given by the rest of the sequence. And in this way, the model learns embeddings that are context aware and have information from the whole protein sequence. Now, for the we can fine-tune these models. The standard way is to update all the weights, but it's not always feasible. A better approach is instead to use a low-rank adaptation, where the original pre-trained weights are kept as they are, and new layers are added in order to represent the low-rank updates to the initial weights. And these updates can enhance some features that are present that are learned already in the initial model, but can make them more important for the specific task considered. So in our model, we apply this low-rank adaptation to the pre-trained models, where we considered two versions of ESM2 and also PROT5 and ANC that use slightly different architectures and also training strategies. And we fine-tune it for the disorder classification on intrinsic and soft disorder. Now, concerning the datasets, for the intrinsic disorder, we relied on DISPROT and CAID, which is the critical assessment for intrinsic disorder challenge. And there have been different versions of CAID. And importantly, they have different ways of labeling residues as intrinsically disordered or not. And we use the NOx definition, which is based on the circular dichroism experimental data. And in all the subsequent evaluation that I will show, we consider for training data only data that were present and were available before the specific CAID challenge considered for evaluation. Instead, for a soft disorder, we created this comprehensive dataset, where starting from the chain labels of the PDB structures, we extracted the high B factor regions, the missing regions, the interface regions. Then we clustered similar structure for the similar sequences, and we computed the union of these classified residues. In this way, we obtained 64,000 clusters that, after sequence reduction and filtering based on sequence identity, we reduced to a lower number for training, validation, and testing. So for the results on the intrinsic disorder challenges, we first evaluated the model on the CAID-1. And what we can see is that all the models based on LoRa fine-tuning outperform slightly the best performing model for that challenge. And in particular, ESM2 based models seems to better capture the intrinsic disorder for the proteins. And this behavior is the same also when we consider the more recent Kite tree challenge. In this case, we had more data for training, so the performance is improved also for the other models, and also many more models were submitted. And also in this case, ESM2 derived embeddings give better performances. For soft disorder, we still have similar performances, but what I would like to highlight here is a comparison with the PLDT, which is usually used as a proxy for intrinsic disorder. Here we computed the correlations between PLDT and soft disorder and intrinsic disorder predictions for those sequences in the test set where AlphaFold structure was available. And what we got is that the PLDT is actually better correlating with soft disorder, which shows how the two concepts are more similar with respect to intrinsic disorder. And as an example of prediction, here we have the results for the SAMP protein, where you can see the AlphaFold confidence of the structure, so the PLDT, the intrinsic disorder, and the soft disorder prediction. And this is interesting when we compare the soft disorder prediction with the union of the interface regions, as I was mentioning before, because when we look at the structured domain here, we see that the two kind of overlap, again highlighting this similarity between interfaces, correlation between interfaces and soft disorder regions. And another interesting aspect is how the attention scores change. So of course, interpret the attention scores inside the model is quite difficult. We did it in an indirect way. So for the ESM model, contact prediction head is already trained, and which is based on the attention scores. And here we computed the differences in these predicted contacts based on the variations induced by the LoRA fine-tuning on intrinsic and soft disorder. And in particular, for the soft disorder, we have a shift in the distribution of the values. Here is a visual example for one protein. So in this plot, we have the variations in this contact prediction probability for the soft disorder and the intrinsic disorder. And in particular, for the soft disorder, we can see this pattern close to the diagonal, where that corresponds to actually two regions that are likely to be soft disorder, in the sense that they are at the end of the sequence and in a flexible region inside the protein. And also, there are other regions where the scores are enhanced, and those correspond actually to contacts between the two monomers. And it's interesting because these two interacting parts of the proteins are linked by a flexible region that might imply a sort of transient interaction. So to conclude, our models predict intrinsic and soft disorder from the sequences, even if there are, as many methods based on protein language models, some limitation on de novo and diverse sequences. Also, these embeddings adapted from the disorder fine-tuning can be used for related downstream tasks as contact predictions or assembly paths. The soft-disk dataset, which is released and is based on the PDB structures, allows to explore this relationship between soft disorder interface and assembly patterns, and also exploring the differences and similarity between intrinsic and soft disorder can help to actually link them to specific molecular functions and maybe also improve our understanding on these disorder-mediated interactions. So I want to acknowledge my supervisor, Alessandra, and our collaborator Beatrice. If you're interested, here is a QR code to that links to the article. And I also want to thank all of you for the attention. Thank you.
We will have some questions now. I can't see very well, but if you have questions, please move to the microphones. Oops, that's our timer. No questions yet. As you think of your questions, maybe I can ask one. So have you tried like an ESM model, starting with the random weights, no pre-training weights? How well does it do in this?
So I didn't, but it's since in the end there aren't that many sequences for training, it's probably going to be much worse.
Okay, there's a question.
Maybe with a small
microphone 2, please. Thank you.
Hey, thank you for the great talk. I'm interested if you were looking for some kind of patterns on the level of the sequence between some kind of a difference between the soft disorder and the intrinsic disorder.
Intrinsic disorder. So on the sequences, not really. Like it's more on how it changes the representation, maybe. And what we found is this kind of patterns. At the level of the sequence, I would say there isn't we didn't run a specific analysis on that, but it would be for sure an idea.
Right, thank you.
Any other questions? I have a question on the data set preparation.
Yeah.
So for some proteins, you will have many PDBs, structures resolved, for some few. Does this affect somehow, does this introduce some bias on the way that you create your labels?
So in part, yes, of course. Like it depends, like especially for the soft disorder, like on how the scores are aggregated. Like we can define different thresholds, of course, for defining the high B factors, and also for defining what is considered soft disorder given n structures and what is not. But in our case, like the distribution of the number of soft disorder regions, of course, depends on the number of structures considered. But on the labelization, there isn't really an effect.
Okay, thank you. Let's thank the speaker once again.
Thank you.